In 1960, he moved from Vancouver to Madison, where he worked on the genetic code, and as we all know, was awarded the Nobel Prize for that work. And while there, he also synthesized a gene for the first time, another really important development in genetics. In 1970, he moved from Madison to Cambridge, MIT, where he stayed for the rest of his life. And while at Cambridge, at MIT, he also again synthesized a gene, another gene, a longer gene than the one he had first synthesized. And most importantly, showed that this gene, chemically synthesized gene put together through enzyme ligases, when introduced into cells, worked perfectly. So this was, again, the first example of a synthetic gene being shown to be functional in vivo. And this is really the beginning of all the techniques that we know about cloning, genetic engineering, and the birth of the biotechnology industry really started from there. So that is really, and then while at MIT, he completely switched from nucleic acids to working on in a different area of membranes and membrane proteins. And there too, over a period of 30 to 35 years, he published some 250 papers and really doing trailblazing work once again, and just phenomenal work in that field. So, and this has lasted until basically his passing away. Now, fittingly for a person of his stature in science, there have been international meetings all over the world, particularly at the universities that he had been. And this is, okay, so fittingly for started with Cambridge in science. There have been in UK in 1985. Then a small meeting, is a kind of an in-house meeting in Cambridge, MIT in 1992. This is just crazy. Okay, and then another meeting at Vancouver, 1993. From 1993, there was a meeting in at Brandeis University. Yeah, this is I am just yeah. Great, thank you. Brandeis University. And then from there was a meeting at Okayama in Japan, 2004, and a meeting in Madison, 2009. And now with the help of Doctors Ganguly and Sharma and Aurora, we now have a meeting also back. The circle is connected back to Chandigarh. So it's wonderful to really have this meeting here in Chandigarh, so in Punjab, where Gobind was really born, and this is where it all started. So with that, then I would like to introduce Marcia Rosner. Marcia was a student of Gobind's, the second PhD student that Gobind had, and Marcia was at MIT. And Marcia was really more or less instrumental, I would have to say, in making Gobind switch from nucleic acids to membranes. So having said that, I would like to invite Marcia to come and give her presentation. And Marcia, I don't have your title here, so okay, all right. So, Marcia.
All right. You can see my title, Rewiring Signaling Pathways in Cancer Cells. So it's really a pleasure and an honor for me to be here and to start the meeting. And I'd like to thank Tom and Rajinder particularly for making this happen. Before I actually talk about the science that I'm doing now, I want to revisit a few memories that I have in Gobind's lab. And the stories that I'm going to tell, I'm actually going to tell particularly for the students in the audience. So the beginning kind of is, how do you become a student of a Gobind Khorana? So when I was a graduate student at MIT, Gobind had recently come, as Tom said, from Wisconsin to MIT. And at the time, Gobind basically had only postdocs in the lab, about 20 of them. So it was a huge laboratory, but no graduate students. And the rumor went around that among graduate students that Gobind actually had it in his contract that he would not take any graduate students. So I remember him actually, giving talks in front of us, and we all went, oh, we don't even have to go because, he won't take us. And so one day, I saw him by the elevator, and I went up to him and I said, Dr. Khorana, is it true that you don't take graduate students? And he looked at me, and he said, I had one 15 years ago. His name was John Moffitt. You heard about him yesterday. He synthesized acetyl-CoA, and he's now vice president of Syntex. That was pretty intimidating. In fact, I thought to myself, I will never even hear about acetyl-CoA again. That's a lie, you'll find out soon. And even more, I have to say this, Tom. Tom just told me yesterday that Tom's thesis project was to synthesize acetyl-CoA, and that John beat him to it. And so Tom had to change the title of his thesis so that he could actually graduate and get a degree. Anyway, so I looked at Gobind and I said, Dr. Khorana, would you consider taking another graduate student? And he thought for a little while, and he said, maybe. Why don't you send me your CV? I didn't have a CV. I was just starting graduate student. So I sent him my application to MIT for graduate school, and he took me on. So, as you can see here, Gobind only worked on big problems. And as Tom said, he had an interesting career. And somewhere in his middle age, it says here, I made a turnaround in my interests, turned out to be good for me in many ways. You can see a little hesitation there. So as Tom said, in the beginning, 1961 to 76, so this actually overlapped my time in the lab, which is around 72 to 80. Not 82, that was all MIT. He was working on the synthesis of a gene. And in fact, many of the people in this room, or a number of people in this room, worked on that. And we, who didn't work on that, in the membrane group, the new group, called them the chain gang. When I came, he was just switching to his interest in membranes. I can't say I'm really responsible for that, but I was part of this initial group that used to sit with him, and we would get together and we would read about it, and we would be really excited. But Goban didn't quite want to move into membranes quite yet. As you can see later, he worked on membrane structure with Parul, who's here. He worked on bacterial rhodopsin with a number of people, and then he finally transitioned to rhodopsin, and you'll hear some talks kind of going off of that. But when I was there, his transition point was working on bacterial lipopolysaccharides. And actually, this was my summer project that became my thesis. Every time I would complain to Goban about this project, and you'll see why in a minute, he would say, there's breadth and depth in every scientific field of investigation, and science is pursued in a variety of styles. In other words, be quiet and work. So what was my project and why? Okay, so the idea was I was supposed to work out the structure of bacterial lipid A, and this is a very Corona type strategy. You take the tools and expertise from one discipline to unresolved problems of another. I think this is what he did his whole life. He had a rationale here, okay? Lipid A is the core of bacterial lipopolysaccharides. Okay, why was he working on this? Because they contained, he thought, a similar phosphodiester bridge as found in polynucleotides. Actually, he was wrong. But I didn't know that for most of my career there. But there was a problem. Lipid A contained a diglucosamine core, and it had O-linked fatty acids. That's not a problem chemically. You can get rid of esterified fatty acids pretty easily. But it also had N-linked fatty acids, and those are very difficult to get rid of. But Goban, undaunted, came to me and said, I've got a solution for you. Slime mold, Dictyostelium, must have amidases that digest lipid A, because actually at the end, if you look at the byproducts, they have this diglucosamine residue. Somehow it disappeared and they eat bacteria. So let's break down lipid A with chemicals and amidases, and we'll figure out the structure. So let me just rephrase my thesis problem. If any of you ever get tempted to work for a Nobel Prize winner, look at this. Unknown substrate, lipid A. Unknown enzymes, amidases, I should purify them. Unknown products. Actually, we sort of had a product, it turned out to be wrong. And then I should find out what the structure was. That was my thesis project. So the philosophy from Goban in doing this was scientific rigor, intellectual integrity, interdisciplinary. It was wonderful training because I had no clue what I was doing, and I tried everything that anybody in the lab ever had working. In the end, I had a thesis which was actually a centerfold in 3 parts. My husband did all the structures. And actually, amazingly, I passed. I actually literally did the thesis in 6 months after 5 years. I actually tried to quit at some point, and Goban wouldn't talk to me for 2 months until I went back. So lessons learned, and I think these are lessons, as you'll see, that I've had my whole career, are try anything, be fearless, and the key is never give up. So I just want to spend a couple of minutes saying what was it like to work in the Corona lab. This is one of the early years when I was there, and some of you may recognize yourselves. I won't point them out, but you can see Goban in the middle. Whoops. Whoa, what did I just do? Okay. So what was it like? One day I come in, Saturday, 9 a.m., note from Goban. I was here. Where were you? So main thing, everybody who's worked in Goban's lab know, you show up and you be in the lab 24/7. You drink hard, mostly Josephine's coffee. I never drank coffee before. I don't drink coffee since. I drink lattes. Play hard, We used to go to the Muddy Charles. We would have beer sessions with kids and dogs. Vermont Citrus, it was great. But most of all, it was just wonderful to be part of this Khurana family. Had such great colleagues, past, present, and future, and I really feel very grateful to have had that opportunity, and also to work with Tom, who is very much an integral part of this group. How has my experience with Goban influenced my scientific career? I'm kind of a bit of an outlier anyway, but I think it's had huge influence. And the first one is, big problem, decided to work on cancer. This is kind of tiny. So at this point, I'd like to switch and just give you a little flavor of some of the things that we're doing in my laboratory. So as I said, the problem I chose was to work on cancer, eventually. And the two actually biggest diseases, at least in the US, are heart disease and cancer. And even though this is kind of an older slide, you can see if I can get this to work, that heart disease, we've actually done a great job dealing with heart disease, in part because actually it's one disease. But cancer has really been largely intractable. It's really been very difficult. So I want to talk a bit about why that is. But the cancer that we focused on is breast cancer. So that's really the major women's cancer. And it's responsible for almost 30% of women's cancers and over 15% of women's deaths from cancer. And in particular, learning from Goban, right? Don't take the easy one, take the hard one. So we focused on triple-negative breast cancer. Those are tumors that are estrogen receptor negative, progesterone receptor negative, HER2 negative. It's about 15 to 20% of breast cancers. It's the most aggressive subtype of breast cancer. There are no targeted therapies for this type of cancer. At least in the US, it disproportionately affects African Americans and lower income women. And it's poor response to chemotherapy or even known targeted therapies. So I thought that's a good one to look at. So why is cancer such a difficult disease to treat? I think the answer is very simple. It's one word, and that it's a heterogeneous disease. It's not one disease. So most cancers have complicated origins. For solid tumors, what actually kills you is not the primary tumor itself, but actually the dissemination or metastasis. And metastasis is characterized by many distinct biological states, and they're actually driven by stress in the environment. So hypoxia, you don't have enough oxygen, you don't have enough nutrients, and so the cells are driven to move out. And down below is just a scheme of a primary tumor moving either through blood vessels or lymph vessels to another site, and that's what's termed a metastasis. So what do I mean by heterogeneity in cancer? It's sort of extraordinary, the heterogeneity in cancer. First, different tissues, as we all know, have different cancers. But then, as I just alluded to, even if you say, okay, I have one type of cancer like breast cancer, there are different subtypes of that cancer. So I told you I'm focusing on triple negative. But then, just take a single cancer in a single person. It turns out the cells are not the same. There's a lot of heterogeneity between the cells within a single tumor. That heterogeneity can be genotypic, so it can actually be in the level of DNA. It can even be phenotypic. You can have the same genotype, but actually a different phenotype, a different set of genes stochastically expressed within those cells. So there's a lot of variance there. Different subcellular signaling pathways. So that's really what I do, signal transduction. Got into the field before they called it that. So you can have many signaling pathways getting you to the same end. So there's enormous complexity. But even more, every tumor is unique. So people now talk about precision medicine, individualized medicine in cancer. But what's most important and usually ignored is that tumors are dynamic, that actually they undergo an evolutionary change with time in response to stress and the microenvironment. So it's this heterogeneity, actually, that prevents treatment. Frankly, we could kill cancer cells, no problem. We would kill you too. Because normal cells basically have the same signaling pathways that cancer cells have. They've just gone awry in the cancer cells. So for those in the cancer field, we really have three goals. We either need to selectively kill cancer cells, or we need to reprogram those cells to eliminate metastatic disease, so that they can now be sensitive to the kinds of treatment we normally use, like chemotherapy. And we have to overcome resistance to treatment, which usually in the end is what kills you. And the major challenge, which I think a lot of people don't realize, is that single drug treatment is largely ineffective. So even though a lot of targeted drugs have been identified, for instance, the poster child is melanoma, there's a particular mutation that is responsible for it, it's called V600 in RAF. If you use a drug to block that RAF, works beautifully. Almost 99% cure for 4 to 7 months, and then bam, it's back. So it's really been ineffective. And the key challenge then is to identify synergistic drug combinations, or now drug combinations in combination with immunotherapy. Some of you may have heard about this. This is really the most exciting aspect, I would say, of cancer therapy right now. And together to eliminate the cellular fitness of cells as they're evolutionarily changing, and eliminate their ability to adapt to their environment. And just to give an analogy, this is sort of what signaling pathways look like in cancer cells, kind of like a hairball. And the analogy for me is like a roadmap. So you can imagine I'm going to go from Memphis to Miami. I've got a nice path. I'm going to use a drug. I'm going to block it. And then whoever's driving says, well, I think I'll go to Boston first. So there are many, many ways of workaround. And so the key is, how do you stop that? How do you actually minimize and block the pathways? So how does this framework inform how we might approach treatment of tumor cells? So this is a strategy that we've suggested, and this is actually a Mac PC problem. Mike and I were saying, wow, great, no problem going from Mac to PC, but this didn't quite work. So I'll just kind of tell you, you can sort of get an idea. Forget that top part and go down here. This is a picture of a heterogeneous tumor, so you can imagine that there's kind of 2 colors of cells there going to a homogeneous tumor, so 1 color. And then you use targeted drugs, and you can actually kill. And the analogy, which I was really alluding to before, is when you have heterogeneity in a tumor, the more aggressive tumors tend to actually have more heterogeneity, whether it's phenotypic or genotypic. And so you can imagine, I'm married to a physicist, so I like to think about things like a physicist. Here's a variance curve, and you can imagine it's a very broad variance curve. And let's say here's the fitness line, and if you're using a drug, you can kill all the cells here, but you still have these outliers that are living. So the goal is, can we narrow this variance? Let's say here, put it in the space where now they're vulnerable, and then hit them with a target drug. So hopefully that's clear, even though the slide is not. And I'll say in a minute, we focused on a protein that we identified called BAC1, where this is where it's expressed, and more heterogeneous, more aggressive tumor, normal aggressive tumor. And this is where we've taken it down. So I want to tell you a little bit about BAC1. So BAC1 is a heme-binding transcription factor. It's called BTB and CNC homology 1. It's a basic leucine zipper transcription factor. And it forms a homodimer or a heterodimer with small MAFs. In general, when it's binding with MAFs, it's actually a repressive factor. So it turns things off, doesn't turn things on. And it regulates genes involved in the oxidative stress response. So I said it's heme binding, so you can sort of see the link, and I'll show it to you a little more directly later. And hypoxia induces BAC1 expression. So I told you that metastatic cells tend to be in a hypoxic environment. So this does not characterize every single metastatic cell, but it characterizes a fair fraction of metastatic cells where BAC1 is induced. So we originally showed that BAC1 is able to promote metastasis. And if you actually suppress BAC1, let's say with shRNA, then, whoops, keep doing that because I don't know where the pointer is. This is an example of a tumor in a mouse, and that's the normal tumor. And actually, this tumor, even though it looks like it's in the brain and in the head, it's actually in the skull, so it's actually a bone tumor. It's a derivative, those who know anything about this, of an MDA231 cell derived by Masakay, so it's bone tropic, and weirdly goes to the skull. if we get rid of BOC1, then you've pretty much wiped out the metastasis. This is a metastatic tumor. You wipe it out, and then we can rescue it with downstream genes. So we kind of worked out signaling pathway for it. Again, those of you who are familiar with signal transduction would know about the MAP kinase pathway, which has really been a focus of a lot of my work. And so BOC1 is down here, and BOC1 is a target of the MAP kinase pathway. So MAP kinase stabilizes MYC, and MYC turns on BOC1, and BOC1 actually suppresses a microRNA. Oh, sorry, I'm in the wrong place. BOC1's here. So MYC turns on LIN28, and LIN28 is an inhibitor of processing of a microRNA LET7, and LET7 actually is a suppressive microRNA, and BOC1 is a target of LET7. So basically, if you activate the MAP kinase pathway, you can lead to activation of BOC1, and that leads to other factors that are involved in metastasis, like matrix metalloproteinases, like MMP1, or CXCR4, which is involved in the ability of circulating tumor cells to adhere to metastatic sites. So it's a metastatic factor, pretty good. Can we learn something about it? We actually went back into human data, and we asked, okay, is BOC1 then more elevated in, let's say, triple negative or so-called basal-like subset of breast cancer rather than non-basal? And the answer was yes, it is, as shown here. We actually, based on that signaling pathway, made a gene signature. And so we started with a metastasis suppressor called RKIP, RAF kinase inhibitory protein. And we didn't have actually LET7 at the time in terms of the targets, so we kind of made up some. And BOC1, we identified some of the BOC1 targets, put them together with some of these downstream guys. And with a gene signature of about 30 genes, we could take, let's say, 871 patients with breast cancer and show that the ones that had this gene signature are more likely, this is actually months, so, sorry, this is years, excuse me. So they're more likely to die. This is metastasis-free survival, so this is normal, and this is those with the gene signature, they're more likely to die. And even those that are among the triple negative, this gene signature can stratify them and find the worst patients. So now we can find patients, but how do we treat these patients? So that kind of took us to the next stage. So we wanted to ask, what are other functions of BOC1, let's say in breast cancer cells? And to do that, we went back to gene expression, just cell lines, and asked, okay, what genes are turned on when you, and what genes are turned off when you get rid of BOC1 compared to control. And here we actually had a surprise, because there was one signature that just kind of was in our face, and that was essentially energy. If it's cellular components, it was mitochondria, and biological processes was energy. So all of a sudden, I'm working on metabolism. I had to take metabolism in graduate school. I even had a professor that made me figure out how all the electrons moved in the TCA cycle. And I thought, I never have to do this again. And there we go, acetyl-CoA. See, I told Tom it's going to show up again. So just to remind you, if you look at energy processing in cells, there are really two aspects of it. One is called glycolysis. So you start out with glucose, end up with pyruvate, and pyruvate can go two directions. So if it's glycolysis, you end up with lactate. A lot of people become lactate intolerant later. This is called the Warburg effect, because in cancer cells, this is actually the dominant way that you generate energy. It's not very efficient in terms of ATP, but it actually gives byproducts to lead to biosynthetic, biosynthetic intermediates, which are needed to make more cells, which essentially is what you do in cancer. Normally, though, pyruvate then gets processed to acetyl-CoA, through pyruvate dehydrogenase, then goes through the TCA cycle, then goes through the electron transport chain. You can see NADH goes to NAD here as one byproduct. NAD is very important for biosynthetic intermediates. And then ATP. So basically, cancer cells used what we call now aerobic glycolysis, primarily up here, but they used some of this as well. So what's BOC1 doing? Well, we were stunned to actually see that what BAC1 was doing was regulating all these enzymes in the electron transport chain. Okay, this is a major process. It's actually suppressing them, so when we take down BAC1, we're inducing them, which is what I'm showing here. And these are some examples of some of the genes that we are looking at. And I'm not going to show you, but actually this happens directly. BAC1 sits on the promoter binding site and regulates these genes. So BAC1's regulating this major biological process. And in essence, just to kind of summarize what we were seeing here, and I'm not going to go through all the details, is, as I said, normally in cancer, glycolysis is dominant and lactate's a major byproduct. You do some oxidative phosphorylation, and reactive oxygen species as a byproduct is fairly low. And what happened when we took down, this is in the high BAC1 tumor, when we took down BAC1, we're essentially shifting the energy balance. So now it's more in the oxidative phosphorylation direction, and we're actually seeing more reactive oxygen species. I'm not going to talk about that, but that's also an interesting aspect of what we see. So now that we saw this, we said, well, I wonder if we can actually take advantage of this in some way. Can loss of BAC1 somehow sensitize cells to inhibitors of oxidative phosphorylation? So have we now shifted the cells in a way to make them sensitive to something that at one point they might have been resistant to? So we turned to a drug which is actually well known in the diabetes field, which is called metformin. So metformin reduces insulin levels, modifies cellular metabolism. One of its targets is AMPK, which is activated when you exercise and you kind of use up all your nutrients. But it turns out it also inhibits the electron transport chain. And in fact, some studies of mice have shown for cancer that if you use metformin, you can actually inhibit tumors, and it can be reversed by putting in a yeast complex 1 subunit, which is insensitive to metformin. So we know that at least one of the major targets then is electron transport chain complex 1. And I just told you that BAC1 actually regulates electron transport chain. And if I take BAC1 down, I'm inducing complex 1. What's interesting is the therapeutic efficacy of metformin is not quite clear. There are actually a lot of clinical trials going on now, but I would suggest that we have no idea which patients we should be selecting. So the results have been mixed, although they're getting more promising. So we said, okay, what happens if we do this, take down BAC1, and then try metformin? So here's a case where we're trying metformin, and we haven't taken down BAC1, and basically, for tumors, this is tumor volume, hasn't done a thing. But now, if we take down BAC1, then actually with metformin, we can reduce the tumor significantly. So this gives you an idea of the shrinkage in size. And this gives you an idea of a graph over time, where this is where we've taken down BAC1 first and then treated with metformin. And this is a case where we haven't touched BAC1, and metformin really doesn't do anything. So that would look pretty good. But, you know, the problem was we took down BAC1 with shRNA. People somehow don't get shRNA the way we treat mice. Actually, we would be done, by the way, if we could experiment on people the way we experiment on mice, but we don't. So we kind of had to say, okay, can we do this another way? Is there some kind of therapeutic way that we can take down BAC1? And amazingly, there was. So it turns out there's a drug called panhematin. It's actually an FDA-approved drug, and it targets BAC1. So just to tell you about panhematin, it's a lyophilized form of an alkaline heme for a disease called porphyria. And it corrects the heme deficiency in liver and represses production of porphyrin precursors. Why does that work? Well, I told you that BAC1 was actually known for regulating redox, and this is part of the reason. Here's BAC1. It actually inhibits an enzyme called heme oxygenase. Heme oxygenase breaks down heme. Heme, when heme is high, can bind to BAC1 when BAC1 is sitting on the promoter, cause it to be exported out of the nucleus, whereupon it gets ubiquitinated and degraded. So this is a very nice little negative feedback loop. If I overload the cell with heme, I can break down BAC1. And here's an illustration where we did that. We added heme, and you can see on the western that BAC1 goes down. This is heme oxygenase, mRNA. So if we add heme, and BOC1 goes down, remember it was suppressing heme oxygenase, then heme oxygenase goes up. So it works in our cells. But okay, let's try this. So can we exploit now heme treatment of BOC1 as a new type of therapy? So we tried this in 2 types of models. One was a so-called xenograft model, where you take a mouse. The mouse is a nude mouse because it has an immunocompromised system. If you didn't do that, then the tumor would be rejected if you put a human tumor into a mouse. So this allows you to put a human tumor. In this case, we used a cell line called MDA436. And as you can see, if you look at the normal growth of the tumor, doesn't really change if you put heme in, doesn't really change that much. Actually, it changes a little bit if you put heme in, doesn't change that much if you put metformin in. But you actually get a dramatic change when you put them both together. And in fact, when we've analyzed these types of mice, we can see that some of them are completely cured. We don't want to say that this is the ultimate cure because, of course, some of them have a bit of a bypass too, but you're definitely doing much better. But right now, the standard is, okay, that's a cell line, you know, it's been grown in culture, not so good. What about patients? So, so we have a new tumor model called a PDX, patient-derived tumor xenograft. And that involves basically a hunk of patient tumors that have been put into mice and never put on a culture dish. They're just transported from mouse to mouse to mouse. So essentially, for a patient, it's their mouse avatar. And so we asked, okay, what about that? Now, these are real tumor cells. What happens there? So here's an example. And this is one where we knew that BOC1 was expressed in the tumor. And so again, metformin doesn't really do anything. Heme does something. Heme does have some other targets, although we have a heme-resistant BOC1, and we could show the effect we're looking at. It seems to be fairly specific. And then again, if you put them together, you get a really dramatic decrease. So just to summarize what I've told you so far, I showed you before, this is the basic component of metabolism. Again, compatibility between PC and MAC. Nice to know that it hasn't totally changed, that they're incompatible. This is supposed to say metformin. So this is the basic scheme I told you. I haven't really shown you, but we know that BOC1 can target also some aspects of glycolysis. We've actually done so-called seahorse experiments, looked at respiration, looked at lactate production, and we know that at the same time that we're increasing oxidative phosphorylation, we're also decreasing production of lactate. So we really are shifting, kind of like seesaw, the balance of the energy pathway. At the same time, we also know, and I really didn't show you, is that we're affecting this step that leads you to acetyl-CoA. See, I can't get away from acetyl-CoA. And BOC1 actually inhibits the ability of pyruvate dehydrogenase to work, so when you take it down, now you're essentially moving pyruvate down this pathway as opposed to lactate. And we also know that BOC1 is even affecting aspects of the TCA cycle, tricarboxylic acid cycle. So, but what I showed you is if we inhibit BOC1 with heme, then we can make the system sensitive to metformin. So that's very well, and that works for breast cancer, and that works in preclinical mouse models. But what about people? Again, we have to kind of go back to people. So can we use BOC1 to first identify patients who benefit from metabolic inhibitors and then potentially try it? And so what we did was go to a new database that's now available to everybody called the TCGA, and there's a few others as well that we've looked at. And it's a great resource because you can get thousands of patients, or a couple thousand patients, and you can look at their gene expression patterns. And then you can actually organize them. You can ask questions about relationships. And I'm going to skip a few things, but what I want to show you is actually an individual patient level. So here's a scheme where you can now plot different genes, like expression, and BOC1 is the top. And going vertically is individual patients. So you can actually ask what's happening with each individual patient. So pink is high expression, blue is low expression, and gray is somewhat in between. And all of these genes down here are different genes within the electron transport chain. So I don't actually have to tell you anything for you to come and say, boy, I think I get this gene signature. When BOC1 is high, electron transport genes are low. Now imagine you're using metformin and you don't have any genes that are expressed to target. It's not going to work, right? So if you're actually taking this whole population, here's a whole population we would predict are completely resistant to metformin treatment. But we think if you actually use heme first, then in fact it would be sensitive. This group here is BOC1 low. And as you can see, I mean, depending on the patient, you get different levels of expression, but overall high of electron transport genes. And we would say, these are the guys that you want to target with metformin. Go for those guys. And these guys are somewhere in between. Maybe we can make them better with heme. Maybe we can find another scheme that takes advantage of the same ideas that we were doing before. So we said, wow, that's great for breast cancer. What about other cancers? I'm not going to show you the pictures, but we can see pictures like this for a number of other cancers. Here's some examples. What we did here was just say something simple. If we look at all the genes in different cancers and we start with BOC1 and just say, what genes are most inversely correlated in expression with BOC1? So here you would say, oh, it's all of these guys because BOC1's high and these guys are low. So we did that, and we asked that for colorectal cancer, for liver cancer, for lung, for skin, ovarian, pancreatic, prostate, and breast, all or breast TNBC. And here we just said, okay, if you take those genes now and ask, what do they correspond to? What are they? The majority fall in the category of oxidative phosphorylation. And in fact, the majority are actually genes in the electron transport chain. So we know that what we're looking at for breast cancer really has application more broadly. And again, we're not getting all patients, but it allows us to stratify them, and we think to actually come up with a new strategy for the most resistant ones. So to summarize, this one actually worked. I don't know why. If we start out with a heterogeneous tumor, then we can actually get rid of one that expresses BOC1, so our BOC1 levels are high. We can reduce BOC1 levels using SI or SH BOC1 or heme or potentially targeted therapeutics. We can get to a more homogeneous tumor state, and that now makes it sensitive to metformin, and we've also done some other treatments. And so we can, some cases cure, some cases get to a much better stage where we're taking advantage now of oxidative phosphorylation, and potentially we can also take advantage of ROS. So we would suggest sort of this strategy for cancer of kind of a two-part, let's go from metastatic, highly diverse and so on, to non-metastatic, more homogeneous, and then let's hit it with more normal therapies like even chemotherapy or radiation. So I want to end with Gobin. Gobin wrote a book, many of you have, which is his selected papers. He worked very hard on that, and he actually summarized each collection of his work. And if you haven't seen this, I would suggest that you do. What struck me in part was kind of the preface. Okay, who did he quote? Louis, and he said, "We must be modest, except in our aims." And I think that really is Gobin in a nutshell. So with that, I'd like to acknowledge the people in my lab who did the work, some very talented postdocs. Casey Frankenberger started it. Jiang Li really did most of the metabolism work I talked about. And Anjin Li was this great undergraduate, physics undergraduate who was flunking out. And I asked him to come to my lab and to start working on some of these computations, and he did all of our gene signatures. He's now a star graduate student at the University of Chicago. So thank you, Gobin, and all of my colleagues from the Corona days. Thank you very much. I don't know, probably we don't do questions, right? Keep going.
You want to answer questions?
Anybody have a question?
Anybody have questions? No? Okay. All right.
One question.
My recollection is that the genes for the mitochondrial electron transport chain are actually in the
My recollection is that the genes for mitochondrial transport chain
Are in the
enzymes
nucleus.
are
You're
in
gonna say
the
nucleus.
mitochondria.
Oh, in the mitochondria. So
And
it's a mixture.
BAC1 is in the nucleus,
It
so I
is.
was wondering
It
how it regulates the expression of genes on a different strand
So there's only
of
about
DNA.
13 genes in the whole collection of genes that are in the mitochondria, actually made in the mitochondria. Most of them are actually made in the nucleus. So, so the specific answer, we haven't actually looked at the individual 13 genes to see whether it's regulating those. We actually looked more broadly at some of the genes that we've seen, and I think the ones we're looking at are in the nucleus. But it's a great question, and actually there's really interesting conversations that go on between them. Okay. So thank you very much.
So, Marcia, okay.
Oh,
That's okay.
Hello, ma'am. You mentioned that BAC1 protein targets LES7 miRNA.
Yeah.
Could you please explain how did you identify the targets?
So actually it's a little bit reverse. So LET7 targets BAC1.
Oh, sorry.
That's okay. How did we figure this out was actually, I think, really interesting. And another example of how strangely I do science, which totally derives from my graduate student days. So what we did was, I mentioned there's a protein there called RAF kinase inhibitory protein, and we knew that that suppressed metastasis. And we also knew by experiments that it regulated LET7, which also suppresses metastasis. It's a marker, it's important for development, a marker in breast cancer. So we wanted to know what the downstream targets might be of LET7. And at the time we were doing it, none were known, actually, none at all. And in the middle of doing it, one just came out, which was something called HMGA2, which we found also. So, so how did we ask that question? It's hard in signaling pathway to go downstream. So went to a colleague. What you do is you actually find smart colleagues, and you get them to do your work for you. So I went to a colleague who's in evolutionary biology, and I said, you study microRNAs. Could you give me 50 genes that you think are likely targets of LET7? A couple we knew, like rats, but most of them we didn't. But if he could find them kind of through evolution, maybe they would be good targets. So he gave me 50 genes. I have no idea why I asked for 50 genes. If I'd asked for 100, this wouldn't have worked. Then we took the 50 genes, and believe it or not, we went to patient data. And the idea was, okay, if you look at gene expression in tumors, and there really is a relationship between RKIP and these downstream genes of LET7, because we couldn't look at LET7 in patient tumors at the time, then it should be an inverse relationship. So every time RKIP is high, these genes should be low in patient tumors. So we looked at like 1,000. And so first we did this for the one gene that was known, which is HMGA2, and as you can imagine, it didn't work at all. It was crazy, totally noisy. You can't get any relationship. But what's really strange is if you take the 50 genes together that I asked for from my colleague, and you ask, is there a statistically significant inverse relationship between RKIP expression and these genes? The answer was yes, amazingly. So altogether, the noise kind of fell out. And then if you then look at the correlations and you take the ones that correlate best inversely, you can actually figure out which ones work best. And BAC1 fell out of that. So that, I'm sure it's a longer explanation than you wanted, but that's how we got to it. Thank you.
Okay, thank you, Marcia. So our next speaker is Mike Gate. Mike came to Govind's lab in the early 70s, Mike?
Yep.
Right? And worked on, worked on the gene synthesis program. And then Mike left to go to the MRC lab, where he's been since then. And so title of Mike's talk is From Oligonucleotide Synthesis to Peptide PMO Conjugates for Treatment of Neuromuscular Disease. Mike.
Thank you, Tom. Thank you, Tom. It's a great pleasure to be here. Thank you very much, Tom and Rajinder, for the great opportunity to pay my respects to Gobind. Gobind was a great inspiration to me particularly for his attitude to solving biological problems by using chemical techniques. And it's that spirit that I worked in all my career, as you will see some of the steps in my career. But it was actually Tom who kept me fairly sane in the lab by talking to me and keeping me calm at times of stress. And because a young postdoc gets quite stressed things. And so I regarded Tom as actually an important mentor in my life as well. So thank you, Tom. Now I spent most of my career at 1975 to 2012 in this building at the MRC, the old building, which was actually opened in the 1960s by the Queen. And my lab was up there, the chemistry lab, which was right next to the fire escape, just in case there were any problems. We never had any problems, but we didn't have to go down the fire escape, but it was right there. But in 2013, we moved into a new lab, actually, on the other side of the campus. It's a wonderful new lab, and that we've been there ever since. And anybody who's coming to England, please come and visit this lab, because it really is an amazing place. I'm now just about to retire, although, as you will see, I'm still going to be very busy, and I shall still maintain an office here, but we're also starting a company, as you'll see. Now, actually, I was, as Marcia says, one of the chain gang making oligonucleotides. This is a picture from 1976, and you will see, where the Gobind, you'll see Rama at the back there. Rama, who's actually here. Now, it's actually Rama who taught me my oligonucleotide synthesis at the time. He was there at the bench right next to me, and I thank Rama. It's wonderful you're here, because you taught me all I needed to know about how to make an oligonucleotide by the phosphodiester chemistry method. And I actually made a sequence in the promoter region here, GGAAGC, GGGGC, actually ingrained in my heart. It took a whole year to make that first oligonucleotide, and then I stayed for a second year to make a second oligonucleotide. So in those days, it took a whole year to make an oligonucleotide. That's why there were so many of us on the chain gang, because it took a long time to make this tyrosine suppressor tRNA gene. But it was functional, as Tom said, and that was really very important. In a series of papers, this is the second gene, the alanine gene, tRNA gene was the first one, but this was the first functional gene in 1979 in the series of J. Barl Chem papers. Very formative period. I made great number of friends and colleagues who have now remained, and Parul is here as well, who was in the lab at the same time. It was a great time. But I obviously moved on, but to Cambridge, and over the period, as Tom said, we had many reunion meetings, and this was the first one that I was one of the co-organizers, along with David Jones, who's actually there. It was a shame that he couldn't come here. He was supposed to be here, and Dick Walker was on the back here. Gobind and Esther in the middle here. And there were lots of people you might, Gordon Tanner was here, I think, and a lot of people you might recognize. Rama was here somewhere here, and Hans Gross, and a lot of Bob Wells, a lot of people at this first meeting here. It was quite an exciting meeting. Gobind also was able to have a reunion with Lord Todd. This is Lord Alexander Todd here. This is the last time that they met, as far as I'm aware. He was just closed his lab now, and he was now a emeritus scientist. But it was a chance for him to meet up, having worked with him as a postdoc some years ago. And also at this meeting was Kell and Ruth Kleppe. Now, Kell Kleppe actually should have got a Nobel Prize with Gobind, and Gobind a second Nobel Prize, because actually the principles of PCR, of polymerase chain amplification of DNA using regular polymerases, was actually Kell and Gobind's idea at the time. Kerry Mullis developed the thermonuclear stable polymerases, but actually the original concept was Kell. But that's another story that I won't go into. But it was we had lots of very important people at this conference in 1985, and of course we've gone on ever since to other ones. Now, when I started in Cambridge in 1975, I decided to stay in oligonucleotides, and I was hired to actually use a new solid phase method. My mentor here was Bob Shepard, a peptide chemist, and we developed methods of solid phase synthesis of DNA using his new polyamide support, which he thought would be freely permeated by polar organic solvents like pyridine and DMF. And so it proved to be. And I used Gobin's phosphodiester chemistry on a solid support and developed the technique of solid phase synthesis. And this was fairly manual, although we used actually a modified Beckman peptide synthesizer to deliver the solvents. It was just purely delivery agent. And the cycle time now was about 24 hours to add one nucleotide. It was quite a lot of work actually in those days, but nevertheless, and there's a series of papers here I published with Bob Shepard here, but nevertheless it was a significant improvement in time. Now it's fairly manual, and Arama recognized the dry box actually that we used to do phosphodiester chemistry, where I still use the dry box in that initial method, and I actually activated the nucleotides with triisopropyl benzene sulfonyl chloride, just as we used to do, and blew them into the reaction vessel. The reaction vessel was held in a little glass, sintered glass funnel, a little glass reaction chamber. And later I used a switching device to switch manually. I modified it from the Beckman synthesizer so I could deliver it by gas pressure. And it had a timer and what have you. So then, after purification, both at the deprotected and protected and deprotected stage, also quite a lot of work. But it actually now took an assembly of about 1 to 2 weeks to make an oligonucleotide by 1979, rather than the 1 year to make an oligonucleotide. So this was already a significant advance, including 1 week for purification. So that was an improvement. This is my colleague Bob Shepard, whose lab it was, and there was his peptide group that I had there. I was afforded little space here, and actually my first assistant was Mohinder Singh, who was actually from Chandigarh. He's actually retired to Chandigarh, and I stayed the last few days with him. He was my assistant for many years, from 1979 to 2003. And there was a couple of other people. There's me at the back there, and Richard Titmus and Hans Mattes were my first postdocs. But these are the other peptide group. It was in the days when we were allowed alcohol in the lab. Nowadays, health and safety don't allow us. This was a party there. But we switched to the triester chemistry method at that point. There was a new method of oximate, a cleavage of the chlorophenol protecting group at that time that Colin Rees introduced. And this inspired me to change to the triester method of chemistry, using first of all a polyamide support and again delivery by the Beckman peptide synthesizer, but later a more rigid Kieselguhr polyamide support. And now we started to pressurize the bottles with nitrogen pressure and pack the resin into a small column. And the small column actually is shown here. These are little columns here, and the resin was packed into. These are 4 columns in parallel. Had a switching device and bottles. And actually, the idea of using pressurized bottles came from Marv Carruthers, who visited my lab in 1981. And suggested, why don't I switch to gas? And in fact, we never looked back. We used apparatus from a small company called Omnifit, which provided the bottles and all the plastic tubing and things like that. And now developed a cycle time of only 1 hour rather than 1 day, and using a long-chain alkylamine controlled pore glass support. We were able to synthesize quite rapidly. So the cycle deprotection afterwards, followed by purification. So now we could assemble very quickly. In fact, my last postdoc at that time, 1984, 1982-84, Brian Sproat, assembled a whole gene by himself on this apparatus, this manual apparatus, using 4-column 23 oligonucleotides up to 22 nucleotides long, synthesized 4 at a time, and the whole synthesis was done, including the cloning and ligation into a vector, M13 vector, and I did that with Mahinder Singh. And the whole thing was done in 3 months. So that's compared to a whole army of people just 8 years previously in Gobin's lab to make all the oligonucleotides and the cloning. So already now things are rapidly improved in time. Actually, we trained quite a lot of molecular biologists came down to my lab from the other parts of the building and actually assembled their own oligonucleotides using this little manual synthesizer. And in fact, the company did rather well. We didn't make any money, but the company sold hundreds of these all around the world, including in India here at the time. So it was actually quite an exciting time developing, seeing our triester chemistry going quite nicely around the world. However, things were changing. Marv Carruthers had developed a wonderful new phosphoramidite chemistry in Boulder, Colorado, and also using a glass support. And he founded the company Applied Biosystems, and that chemistry was just slightly better than the phosphotriester chemistry. It gave a higher cycle yield, and therefore you get a longer oligonucleotides quite quickly. And because of the starting of the company Applied Biosystems, very, very good engineering, it was clear that the race to make oligonucleotides and automate it was actually run by Marv and his company. We switched to the phosphoramidite method in 1985 and never looked back. This is me with some hair at that time, posing in front of the machine. I didn't do it. It was a whole series of people. We formed an oligonucleotide synthesis service in 1985, and that lasted to 2010 with a whole of these sorts of people. And we could make whole genes, and this was actually a collaboration with Pushpa Bhargava in Hyderabad, making a seminal plasmin gene in 1987, which we made and expressed the protein and did some protein engineering with. And this was, of course, done quite quickly. The oligonucleotides can be made now in just a day with no problem at all. So having the oligonucleotide synthesis was now essentially solved in various other methods, and particularly looking at the hairpin and the hammerhead ribozymes. It was actually Aaron Klug who asked me to start thinking about working on the hammerhead ribozyme. He wanted to actually crystallize it. And so we started to look at that, and I decided to do some chemistry on this. And we first of all worked on the hammerhead ribozyme, and we were the first to show using configurationally defined phosphorothioates. Phosphorothioates came from Fritz Eckstein's lab, to study the mechanism of cleavage of the hammerhead ribozymes, and showed it actually was, as predicted, an inline cleavage of the hammerhead ribozyme. And then went on to the hairpin ribozyme and did some work on the structure, and putting in some base analogues, looking at the purine functional groups, and then later doing some cross-linking here, looking at the structure here. We worked with Frank Zahler. Eric Westhoff did some modeling for us, and Fritz Eckstein provided some materials for us. And we're actually the predicted structure of the hairpin ribozyme turned out to be very close to that when the crystal structure came, so we were quite kind of happy about that. And we also looked at aminoglycoside antibiotics and polyamine spermine, showing we could reduce the metal ion requirement for the hairpin ribozyme cleavage. So that was quite an informative time and quite exciting. But in a change of pace occurred in 1988 when I was looking for a more a mammalian system to work on. I thought it was time to go from, I'd done some cloning in bacteria. We expressed RNA ligase and cloned it. That was a whole other story that I don't have time to go into. But I thought it ought to go into now, it's time to go into mammalian biology. And just at that point, the HIV program was starting up, the AIDS-directed program in the UK. And I started working with Jonathan Kahn, another scientist in the MRC lab, on the HIV, TAT, and REV proteins, which are important regulatory proteins there, and their interactions with RNA substrates, and their binding proteins. Not much was known about that at the time, and we were able to use synthetic chemistry to synthesize the RNAs, sections of the Rev response element and the TAR RNA, synthesize the proteins as genes and express them. So we made synthetic genes in the lab, expressed the proteins, showed the binding, and actually that was our first paper in the EMBO journal, in the TAC protein stimulates transcription by binding to the TAR RNA. And we did some mapping of contacts between the Rev response element and the RNA as well. So quite a lot of RNA protein interactions, looking to see if we could develop drugs against these specific targets. The whole series of postdocs in the lab here. And John Kahn then at that point founded a small company called RiboTargets in Cambridge to look for small molecules inhibitors of these protein RNA into targets, and also on the ribosome. There were others who joined us, including Venki Ramakrishnan at the time, joined us in this RiboTargets company. However, I wasn't never clear that small molecules would be good for these RNA targets. In fact, it proved to be that it was proved to be much more difficult to define small molecules which would stably interact and block these RNA protein interactions. So the company eventually folded or was taken over by another company. But the idea for me was not small molecules, but oligonucleotides and the antisense area, which to actually block protein RNA interactions. The idea of antisense goes back to Paul Zamechnik back in the late 1970s. And he had the idea to block such interactions by a steric block mechanism. But all the companies which had started in the mid-1980s had gone on to the regular antisense mechanism, which was cleavage by RNase H. However, I thought this was an ideal target to look at steric blocking oligonucleotides, and I teamed up with Jesper Wengel's lab in Denmark to look at locked nucleic acids with 2'-O-methyl nucleotides, and also PNA from Peter Nielsen's lab, also in Denmark. And so we collaborated quite strongly here, and were able to design short oligonucleotides which interacted here, opened up the RNA sequence here, and blocked the binding of the TAC protein, and got actually interference with the transactivation mechanism. And this was a kind of a formative period, showing that it was possible here. We also did some work on the ribosome using similar short oligonucleotides. So we started to work on this, but it was clear that if we got some very good results in cell-free systems and showing blocking of transcription. The problem was now, how do we get these oligonucleotides into the cell? Because this was the key issue which antisense companies were having problems with in the 1980s. And by the mid-1990s, actually, quite a lot of work had stopped, and several companies had given up at this point. And so I thought this was a challenge in the latter part of my career to work on delivery of oligonucleotides into cells. And we started to think about peptides. Having spent some time with Bernard Lebleur in Montpellier, I decided that this was the opportunity to think about delivery using peptide conjugates of oligonucleotides, using peptides as delivery agents. And there was a so-called cell-penetrating peptides, which was starting to be developed at this time. And so Suzanne Perrotte, also from Montpellier, was my first postdoc in this. And I had a series of postdocs looking at the chemistry of joining oligonucleotides and peptides chemically, and we had various techniques here. The simplest turned out to be a simple disulfide joining, and John Turner in my lab developed the chemistry for this. It was actually simpler than anything else. And we were able to make oligonucleotide peptides and show transactivation. And we showed a small amount of activity inside the cell using the sorts of HIV, TAT, and penetratin peptides by this technique. Now, just as a sideline here, 5 minutes, okay. Just as a sideline here, we did some work on microRNA and showed that actually we started to work with quite a lot with PNA. And we showed that microRNA was not we did not require a cell-penetrating peptide to target microRNA. We just needed a few lysines to get inside, and this showed that the microRNAs were not were very accessible and not like the nuclear targets at all. And we did quite a lot of work with microRNA 122 and 155. This was in liver and this was in spleen, but both in cell and also in vivo, showing that the possibilities of using PNA as an oligonucleotide target. So that's just an aside. Going back to the main project, we started to use oligonucleotide peptide conjugates for delivery into cells. And we particularly used, we started with regular oligonucleotides, but actually we got much more success with PNA, with charge neutral oligonucleotides. And particularly with a peptide, this was a transportan cell-penetrating peptide, which gave a little bit of activity. But as soon as we added 6 arginines on the end of it, we had massive activity. So suddenly, the arginines allowed us to deliver inside the cell, and particularly of peptide nucleic acids, and we used not only an HIV target, which was our first one, but we started to use Richard Cole's splicing model, because it actually had an upregulation and a much bigger dynamic range in which to use our techniques. And so we were able to block splicing here, and using his luciferase model, we were able to generate luciferase, upregulate it, and using R6 penetrating without any transfection agent, just incubating with cells, and we're able to get into the nucleus and increase the luciferase, showing that we could now have a mechanism of splicing control using these peptide PNA conjugates. Now, I just want to tell you a little bit briefly, but we then teamed up with Matthew Wood in Oxford to look at muscular dystrophies. These are very horrible diseases of young children. The frequency of about 1 in 3,500. This is Duchenne muscular dystrophy. Patients are in wheelchairs by age of 12. There's tremendous degeneration of the muscle fibers. And what dystrophin does, it's a very important protein that anchors actin filaments to the sarcolemma membrane and muscle fibers. And in Duchenne muscular dystrophy, this is a truncated, there's a genetic deletion here which truncates the protein. So you actually get deletions of point mutations. And it's a very long, stringy gene of 79 exons, and all have to be spliced correctly. And that's what happens. Unfortunately, you don't get dystrophin produced, and you get muscle degeneration. There's Becker muscular dystrophy, which is a benign phenotype. Certain in-frame deletions leading to normal life expectancy. And so Hans van Ommen in the Netherlands had this idea of converting Duchenne muscular dystrophy to Becker muscular dystrophy using an oligonucleotide. For example, if you've got a deletion in exon 50 here, you can't get exon 4950, the correct splicing. So you jump over to the next one, exon 51, and you get an outer-frame messenger RNA, and therefore no dystrophin. Whereas if you can block that splicing event here by an oligonucleotide, then you can jump over that to make an exon 4952 fusion, which is now in frame, and now you get the Becker muscular-like dystrophy, which is very, very mild symptoms. And people have now developed drugs for these, initially 2'-methyl-phosphorothioates, but unfortunately those were not successful in the clinic. Tricyclos are being developed by Christian Leumann, which are looking very popular, but the major ones are peptide morpholino oligonucleotides of Jim Somerton, also about 30 years old, as well as PNA is also about the same. And recently, atiplacin, a 30-mer oligonucleotide against exon 51, just the correct sequence, has got FDA approval in October 2016. I like PMO because it's stable to nucleases and proteases, like PNA. It forms strong sequence-specific duplexes. It's non-toxic. It's excreted from the body completely whole, and it's shown in vivo biological activity in many models. And in particular, we can couple to it, just like PNA, some peptides. We did this starting with our R6 penetratin peptide, which we developed before, and now trying to make it protease stable. We introduced some aminohexanoic acid derivatives and some D residues to form our first PIP peptides, PNA PMO internalization peptides. We've published all of this, and these initial PIP peptides showed a higher number of dystrophin-positive fibers than the competitors, the Sarepta companies' peptides. And our collaborators, Matthew Wood and colleagues here, who developed this, we're rather excited that we could now, in vivo, get really good results and delivery with our peptide PMOs. And we first of all had PNA, and then switched to PMO, because actually PMO was much better in muscle rather than PNA. And we've gone on to look at the 5 series. We can now simplify this, so there's aminohexanoic acid and arginines on each side, a short hydrophobic section in the middle. And the exciting thing was that the short hydrophobic region gave us much more activity in heart, whereas arginine-rich regions in general, I will need just one more minute, was very good for all muscles. Heart required this hydrophobic core, and we were able to show that this hydrophobic core is not sequence dependent, but was just needed a hydrophobic core of about 4 to 6 amino acids. These now became important delivery mechanisms, but in vivo, by IV injection into animals. In particular, we have this dystrophic animal called the MDX mouse. And we've gone on to try to develop this into a drug. First of all, with a large company, we licensed this to a large company, Shire Pharmaceuticals, and we developed slightly smaller peptides, PIP9B2, for example. There was several of these here. However, unfortunately, the company changed management and hasn't developed it. So we were very upset about this. So we decided to beat our own patent and develop our own peptides. And so since then, we've been developing our own peptides, which are not peptides, very similar. I can't tell you about their sequences, but they're arginine-rich sequences, again, rather similar. But we're looking now for better tolerance in the mouse and the rat. It's also driven by toxicology now. And we're looking for various high therapeutic index peptides. And we've now got a series of these. And we have been able recently to show that we can get high activity with low effectivity in kidney technique of delivery into the mitochondria. We are developing this as a drug. This is my final group here. Andre and Frank are my last 2 postdocs. I have a collaboration with Matthew Wood. I shall be working very strongly with Matthew Wood to develop these compounds. And we're starting a company in Matthew Wood's lab now to develop these. A company we'll call PepGen, to bring peptide PMO to the clinic. And I just want to thank you now. Many co-workers have passed through my lab, and also to my heritage of Govind Khurana and Tom for setting me off on this trail. And thanks to many recent funders and supporters here who have worked with me. And thank you very much for your time. Sorry, I'm just a little bit over.
Thank you, Mike, for this very interesting work. So most of it is really nuclear targeted, right?
Yes, the nuclear targeting is the most important, and getting things into the nucleus requires a peptide, and it requires an arginine-rich peptide to get there. Targeting microRNA is very different. There you can target without requirements of a peptide. But here, it's the peptide delivery, and this is what we're now concentrating on, finding peptides which are short enough and with fewer arginines as possible, we can keep the toxicology down in kidney in particular as an issue. But yes, sufficiently high enough activity in mouse models. So we're quite excited about this, and we think we can bring these to clinical trials.
Great. One question. Somebody. Yes.
Yeah, it's a very interesting question. We actually don't know how it crosses the blood-brain barrier. Probably the arginine-rich peptides use absorptive transcytosis. We don't think it's a receptor-mediated process, although we're looking at peptides which also have a component involving receptor-mediated. But these we think are not receptor-mediated, and we don't actually know precisely how it gets across the blood-brain barrier, but it certainly gets into the spinal cord and into the brain of these mouse models, and we've shown that in a couple of examples published quite recently with one of our PIP peptides and another peptide. And we're now developing this new class of shorter peptides here. But so we're not. It's very difficult to do experiments in this area, but we think it's an absorptive transcytosis. Sorry? Percentage getting into the... It's very difficult to assess that. It's a very small amount. A very, very small...
Okay. He wants a quick question.
Yeah. No, there's no signal. Basically, you get released. What happens is you go through an endocytotic pathway taken up by one of the endocytotic mechanisms into the early endosome, then into the multivesicular bodies, late endosome. These things are then released. The arginines, the hydrophobic components, help to distort the membrane, the endosomal membrane, allowing leakage, and then it automatically goes in the nucleus. Yeah, correct. They'll perturb the membrane. Yeah. Yeah. You don't need anything to go in the nucleus. It's small enough. If your oligo is small enough, if you have a very long oligo, 50 or 100 long, then you need a transporter system. But these are short enough to just go straight into the nucleus. There's no barrier once you get into the cytosol.
Okay, lunchtime. You can talk to him. Okay.
Okay.
All right. Our next speaker is Asim Ansari. Asim is not a member of the Karana alum in the strict sense of the terms, but he is an honorary member and most honorable member of the Karana team. Yeah. So Asim has done a lot for the Karana program. He started in Wisconsin the Karana internship program that allows lots of students from this country to spend a summer in the United States with the help of the Indian government. So Asim is just done a wonderful job there, and he's expanding into all kinds of stuff. He might even say a few words about it. But today I think what mostly is give a talk on creating synthetic genome switches to control cell fate. Asim.
So it's an honor and a pleasure to be here. I had never trained with Gobind, but I met him briefly with Tom. And I have read his papers and I took inspiration from his words. So it's the Eklavya model of training. Many of the Indians here will know what that means. From a distance, he trained me to be a good scientist. So what I'm going to tell you about is how that fusion of bringing chemistry to biology has influenced my own work. The talk is in two parts. 2/3 of it will be about making precision therapeutics that Marsha, wherever she is, where's Marsha? Yes, Marsha disparaged. So I'll make a case for it. And the third of the talk will depend upon the timing. It might go faster than a third. But that'll be about the Khurana program, where we are planning to take it and what the next steps might be, and seeking your advice on how it should be crafted as we go forward. So this is the topic of the day for my talk anyways. Let me start the timer because I saw what Tom did to speakers who are a little longer than they should have been. So I'll talk about transcription factors as we understand them chemically. So people know transcription factors control gene expression. If it moves forward, we'll see. Oh, it's not moving. They have to point it there, I see. So these are the 3 areas that my lab works in. We try to understand mechanisms in model organisms to try and figure out how the machinery actually works. Surprisingly, it's still being determined. We make synthetic transcription factors. I'll tell you more about that. And we also try to make synthetic genomes altogether. So here, we try to understand how signals, just like Marcia was describing in her very elegant work over the years, how signals lead to activation of transcription factors in this case. So she talked about BAC1. This is a more generic version of that idea that there is some molecule, protein, that recognizes that it is now to go find its place in the genome. Once it gets to that place, it either turns on, which is how we typically think of transcription factors, but also in the example of BAC1 with maths, it turns off. So how you regulate the genome, what genes you target, how you turn it on, how that specificity is defined, where is this factor supposed to go? We spend a lot of time trying to understand that. Once it gets there, how does it engage the machinery? We spend a lot of time doing that. And most of these studies are done in yeast, bacteria, Drosophila, model organisms. And that has influenced how we go about doing this, which is making so similar or artificial transcription factors, or ATFs, that can target sequences that you're interested in. So you now, by design, have made molecules that can target a specific site in the genome or multiple related sites. And by making those molecules and conferring on it on or off switch capabilities, you can now drive genes. And by driving specific genes and their networks, you can drive cell fate. Right? So that's a part of what we do. We've taken cells that are fibroblasts, converted into stem cells. We've taken stem cells, converted them to heart, neuron, pancreas, etc. Beta pancreatic cells. So that's one part of my lab that's focused on. Beyond this, this is protein-based. You've changed the tips of this protein to target a specific site in the genome. You've changed regulatory elements to turn them on or off, proximal genes on or off. We've taken the next step to make these things completely synthetic. But let me just finish the idea. Beyond transcription factors, so these gene switches that can target sites in the genome, turn them on, off in a controlled way, can you make an entire new genome? Right? So you heard some really elegant work described by Dr. Gate right now. And yes, there you are. And Mark Kruder's chemistry, using that exact same chemistry with a photoprotected group and that projector, in fact, the digital micro mirrors that are in that projector, you can on a glass slide synthesize 6 million different oligonucleotides, 100 nucleotides long. You can make genome scale genomes in a matter of a night. And then we have attached to that microfluidic devices where you can collect similarly sequenced genes, sequences, separate them out, assemble those things into larger fragments, make things up to 16 to 600,000 base pairs in a matter of a week. Completely clean. So that's where the world is going. So you heard about a gene being made in 3 months. You can make a genome in a day or two. If you're at one of these big places, in my lab you can do it in a week. So that is where we are in my lab on the whole. But this is what I want to tell you about today. So this is truly bringing chemistry to biology. And I thought this would be the audience to tell the story. And this is, of course, inspired by the way Gobind did chemistry. Long before Stuart Schreiber named chemical biology, I used to tell him there's this book by a man called Gobind Khurana. He's just down the street. And he used to smile and say, but Haseem, this is different. I don't think so. My time's up already? How much time have you given me? But basically it comes back to the ideas that are borrowed from biology. Right? So people have been looking at natural transcription factors for a very long time, since the 60s. And what they've determined, people with whom I trained, have determined that basically you need 2 bits of information. One bit of information is a DBD, DNA binding domain. It'll take you where you need to go in the genome. So it's conferring specificity as to location on the genome. The second bit of information you need is the interaction domain. At least I call it an interaction domain. Other people call it activation domains, repression domains, effector domains, etc. Multiple names. But the idea is that you're going to bring in the machinery to do something interesting. So if you have those 2 together, you should be able to turn on a gene. And what we've done is we've taken this thought and made synthetic counterparts. No protein here. It's a completely synthetic counterpart that has those 2 bits of information. And we've taken that information and targeted it to a disease called Friedreich's ataxia. This happens to people, leads to scoliosis, then leads to shutdown of the heart, and then loss of cerebellar material, so people can't walk very well, cannot control their motor muscles and neurons. And so basically you have ataxia. Ataxia means you cannot walk, you have trouble walking. Ataxia of this kind actually emerges because a gene called frataxin right here is not being synthesized. We talked about mitochondrial genes. This is a nuclear encoded protein that then goes to the mitochondria and functions there in electron transfer chain, doing the iron sulfur clusters that are required by the electron transfer chain. So there's a connectivity to what Marsha was just talking about. So how does this happen? Why does frataxin go down? So the normal gene, frataxin gene, has 5 exons, and it has between the first and the second exon a GAA repeat, a trinucleotide repeat, typically under 30. Everybody sitting in this room has at least one allele, meaning one of the 2 genes that you have from your father or your mother, that has less than 30 repeats. If you have more than 30 repeats, this is what happens. Your transcription levels, your levels of FXN RNA, frataxin RNA, go down, and you develop Friedreich's ataxia. Depending on the number of repeats you have, it happens sooner or later. And you have to have both alleles hit in this way. If you have one, you can be a carrier and you'll be just fine. Make half as much protein, you're fine. However, people have been studying this disease because it's one of the most prominent recessive ataxias that are inherited. It's a monogenic disease, meaning one gene leads to this disease. So if you can fix this gene, you'll fix the disease. Along the same lines as Dr. Gates' arguments with muscular dystrophy, etc. The understanding of how this happens might lead to a rational way of tackling it. And so people have studied what this GAA repeat does. So now the GAA repeat, on one allele you have 650 copies of it, on the other one you have 1,000 copies of it. And this particular person, that's his brother, you could easily watch how that lack of that gene affected the motor functions, then affected the heart, and the brother eventually died, but the cells were retained, and we've been working on these cells. And so what people have done is actually try to see compare these two, what is happening here? Why is it that you're not making this RNA? So here's one model, a lot of very beautiful work done here in Delhi and in Chandigarh, led to the argument that this sequence can form unusual DNA. It can form triplexes, it can form R loops, it can form sticky DNA. And the polymerase, this is the RNA polymerase that is transcribing that gene, gets stuck. It's trapped in that state. The other models that have existed out there, become more popular recently, are more driven by chromatin, so that this sequence here somehow leads to wrapping up of this DNA in a repressive state. So DNA is wrapped around nucleosomes. Those nucleosomes are marked with repressive marks. Those repressive marks are recognized by proteins, and the DNA is condensed. And now polymerase has trouble getting through. Both these groups, and they sort of argue that perhaps this leads to that, but there's no clarity in terms of which one of these mechanisms is blocking transcription. But the evidence is clear that transcription, in fact, does go down, and a lot of people have validated that that is true. So what we decided to ask is, at what level are those repeats inhibiting transcription? Right? There are some people who argue that polymerase can't load at all, and so what we did is a simple chip experiment. This is where you throw in formaldehyde, you cross-link the protein to DNA, you pull out the protein of interest, in this case RNA polymerase that is going to transcribe that gene, and you ask, is it there at all, first of all? Second, if it's there, where is it? Is it at the promoter? Here's where the gene is starting, and the answer is not quite. It's left the station. It's made the first exon. Is it at the repeats? Is it trapped in the repeats, as the model for sticky DNA suggested? Not quite. It's not there at all. In fact, it stops well before it hits the repeats. All right? So we know that there's a stalled polymerase there, and it has trouble getting through the repeats. And the work was done by Matt Lawler at Harvard Medical School and my students. So the idea is, the simple idea that we decided to pursue, is that here are those GAA repeats. They are doing something to shut this polymerase down, prevent it from going through, but they do not prevent its engagement at the promoter and initiation of transcription. So it starts just fine, hits the end of the first exon, and then has trouble getting through. The idea is that can we make a molecule that can dock into these GA repeats in that way, so this part of it will recognize GA and deliver an interaction domain, through an interaction domain, the elongation machinery, something that will allow the polymerase to get through this repressive region. Okay? And so if it does, so one half of the molecule is going to recognize, this part is going to recognize GA, the other part is going to deliver the elongation machinery, and that is going to act on polymerase to mark it in a way that it can collect friends and family that help it along through the repressive region, come out on the other side, and follow all the necessary cues that are in that gene to do what it needs to do, splice, terminate, and then let the transcript be exported and converted into protein. So you have this two modular molecule that we are going to make, bifunctional molecule. One module has to be DNA targeting. These are the molecules that are currently used. These are the strategies that are currently used commonly to target sequences in the genome. The CRISPR-Cas approach is incredibly easy to use, intuitive. Even high school students do this easily. So this has become quite popular very rapidly. This perhaps is the more stringent way of targeting the genome. These are proteins that can be designed to target single nucleotides, and they are very precise, much more precise than this or anything else. Zinc fingers used to be the way people used to target the genome. There are many complications because of fingers talking to each other and influencing binding. And then there are these small molecules that are derived from distamycin, versions of distamycin, that can sit in the minor groove, not the major groove, sit in the minor groove and drive and interfere with transcription, but they can be modified in a way to target specific sequences. So this is the size of the molecule, and this is, if you were to draw the chemical structure, what the molecule looks like. So Peter Derwin, Lown, Wemmer, a lot of people spent many years trying to understand how distamycin, which is just these three N-methylperols attached with amide bonds, bind DNA. And it turns out that this sequence is made by bacteria, and it targets AT-rich sequences, and it keeps bacteria with AT-rich genomes from being replicated, from growing, sort of holds them. It's a chemical warfare, if you will. They're preventing their enemies or neighbors, if you will. Typically it's the neighbors, isn't it? Who are your worst enemies? So they tell me. In any case, so what Derwin and others have done is replace these rings with other types of rings. So here you have a hydroxymethylperol, you have N-methylimidazole, and these are just some of the examples. Many, many rings have been used because it's such a modular system with a peptide bond, with an amide bond right there. What they've also done, people who've been developing this field over 20 years, is attach two molecules together with a flexible tether, and when you stack them, they fit in the minor groove quite snugly, and when they are fit that way, they tend to be very specific. A stack of N-methylimidazole and an N-methylperol almost always takes you to a G base pair. If you flip the orientation, you target a CG base pair. If you have a perol against a perol, where is it? There it is. You can now tell the difference between A and T, but you can certainly tell the difference between a C and G. So these sorts of modular recognition rules that were developed by Derwin and colleagues, many colleagues in fact, have been codified in this manner, and we use that to figure out how specific are these molecules. We made a variety of molecules, we tested them against single sequences, then we realized that that may be misleading because they may bind other things we just don't know. So we made microarrays that had millions of sequences on them, every possible 12mer represented on the array as a duplex, so that it's an unbiased screen of what the molecule likes. You label the molecule, you put it on the array, etc., etc. 10 years of my life was spent doing that sort of thing, right? And in the end, my student said, but how does it work in a cell? And that's completely changed how we think about this. So here, Graham Irwin, who is now at Stanford, basically did the simple experiment that we do with proteins all the time. You have a cross-linker, you cross-link proteins to DNA, just like I showed you for polymerase, to ask where is the polymerase? It's past the start site, you can see it very clearly. So you ask, where is the small molecule? Now it turns out, unfortunately, and we could have predicted this just from looking at the chemical structure, that formaldehyde is not going to do it for us. Now my colleague at Caltech, Peter Derwin, soldiered on. He attached various side chains to ensure that perhaps you could just use the same approach, formaldehyde cross-link, come in with click chemistry, pull out the compound and the DNA that is cross-linked to that compound. And they spent a lot of time. What Graham did was a little less elegant. He decided to go get psoralen and a biotin. So biotin is a affinity tag, if you will, that is quite robust, 10 to the 15th, if I'm not mistaken, KD. And psoralen is a planar molecule that intercalates between AT base pairs and does a makes a photoadduct depending on the wavelength that you shoot it with, di or mono. A bulky molecule, he made sure that there were linkers so that the cross-linking module could go find wherever it needed to dock. We did the calculations, what is the frequency of AT sequences in the genome, etc. Based on that, we came up with this model. And this is very different from what my colleagues in the chemical world have been focused on. The idea is that here's that small molecule that I've been showing you. Here's a crystal structure actually of that small molecule bound to the minor groove, bound to DNA that is wrapped around the nucleosome by the person who did the structure of nucleosome, Carolyn Luger. So she did these structures, and you can clearly see the molecule can bind nucleosomal DNA. But what we did not expect, that it would actually bind clusters of these sites. So there are multiple sites, and red means strong binding, really bad binding, sort of exemplified here, that the molecules can bind as a cluster, and that is where you really find them. If you have a single strong binding site, and that has been the, the brass ring, the challenge that everybody has chased, can I make a molecule that will target one site in the genome? And this is perhaps the lesson. Looking at how biology does it, the one thing that we do, we try and see how transcription factors do it. And looking at the way, people think about it from chemical perspective, like trying to dock something into an active site, a unique location where a molecule can be fitted just so. Those two ideas don't quite fit when you're faced with a genome. You have many local minima, and the way natural factors do it, that they sample the genome, and they tend to stick around where there are other proteins that stabilize them. There's a couple equilibria that holds them there. As the dwell time increases, these things can do their biological function from these locations. There isn't one transcription factor that functions only at one site, ever. All right? And so we brought that biological thinking back, and that data was telling us that our molecules were doing exactly that. And so we started using this idea, which we call summation of sites, to go look through the genome to see where these molecules would go. And what we found, of course, we came back to this Friedrich's ataxia model, because what you have here is clusters of GAA sequences. So we made molecules that will target those GAA sequences. We attach to it a molecule that will dock into a specific protein that brings in the elongation machinery that will allow polymerase to get through. So here is the DNA binding module. Here is the interaction module. The interaction module actually looks like this. It's a molecule called JQ1 made by a postdoc of an undergraduate of mine, who's now the head of Novartis. So JQ1 is Junqi1. Junqi1's molecule allows us to conjugate at that end a PEG linker attached to our polyamide. And basically, the length of the PEG linker is such that it has to permit the binding of these two modules into their respective biomolecules, the nucleosome and BRE4, BD1 domain. These are the molecules that we made. Here's the linear architecture polyamide. This is a hairpin. This is a linear molecule that targets GAA, GAA, GAA. All right, so that's the representation of that. Imidazoles are colored in. And so you have just the plain imidazole, or you have, after a certain linker, here's the PEG linker that we used, is JQ1. So in any case, so now we have this conjugate, which we call SYNTEF1. And we have another one that targets a different sequence. There's the HIF1 alpha targeting site, also attached to JQ1. And so here's this story just appeared 2 days ago in Science. And so here's the test. Here's the test where we now take the molecule and we ask, where does the molecule go? The molecule, in fact, does go to those GAA sites, the repeats in that particular cell line. We ask then, once it gets there, does it bring in BRE4? The answer is yes, it does bring in BRE4. Here you are. This is a cell treated with the polyamide alone. This is a cell treated with the polyamide alone. And this is a cell treated and JQ1 independently. They are not conjugated. But when you conjugate them, now you can very clearly see that BRE4 is being brought in. This elongation module is being brought in right over those repeats. And it seems to be interacting with something upstream where there is no compound. So we argue that perhaps this elongation machine is talking to the stalled polymerase through other partners. And there's evidence for that there are in fact proteins that mediate that interaction. And you can see that interaction then leaves polymerase. Here's polymerase. That was BRE4. Polymerase now escapes from that site and goes into the gene. And what is even more compelling than the little bit of polymerase that you see get into the gene is that when it does that, associated with BRD4 is a kinase that is critical for elongation called positive transcription elongation factor B, PTEF-B. So this thing here phosphorylates the polymerase on its tail And that phosphorylation is absolutely important. It's a mark, it's a signal that, okay, now bring the machine that will allow me to get through this part. And that mark is unmistakable, right? So when you have the compound, here's the repeat, here's that little bit of polymerase, but it is modified in a way that it should be modified. And what is even more gratifying is that as the molecule, as the polymerase transcribes right past these repeats and into the gene and then all the way to the stop sign, you find that there's another peak there exactly where it should be. And our molecule is not working at that distance, but the polymerase is using the cues in the gene to do what it needs to do to make the functional transcript. So here's the functional transcript being made in a diseased cell. Here's the functional transcript not being perturbed significantly in a healthy cell, right? So you can see that as you give more compound, you make more restore function to almost 100%. You see that the other molecule does not do this. And not only that, when you look at the entire transcriptome, all RNAs in the cell, in this diseased cell, the one thing that is being moved significantly and maximally is the gene that we are targeting. So it's not affecting a lot of different things. And in this case, where there's a healthy cell, the same compound is doing virtually nothing in this volcano plot. You make the RNA, you splice it, you make the protein, you process it to the size it needs to be, and you do not perturb anything in a normal cell. So we have a molecule that can now reverse the problems that you see with frataxin. The question is now, does it do anything beyond that? It is a small molecule, and I just told you earlier that it samples the genome in many places and sticks around in many places to different levels. Right? The dwell time at different locations differs depending on the affinity and number of binding sites. And so here we are ranking them by order, 250 locations in the genome where this compound can go. So it's sampling everything. It stays a little longer at the frataxin gene, but it stays long enough to bring in the partner, BRD4, to those sites. And yet what we're seeing is a single gene is moving. If there are 250, let's assume that this is at least half of them. There should be 125 genes that have moved to some extent. And when you draw these lines, there are only 11 genes that move. Half of them are in response to the fact that you've restored frataxin function. Okay, the other half, you don't know what they are. But in any case, when you look a little closer, this is what you see. Before you treat with the compound, you ask, where is polymerase? And you find that there's only one gene there, but the polymerase is anchored at the start site. There are a couple others, these are already on, and a compound does not change them that much. And it's only one of them where there's a change in serine 2 levels. This is the phosphorylation that tells you you're going to transcribe through. And so basically what is happening, even though the compound is bound to 250 different places at different levels, it is only acting at that one gene because there's a receptive polymerase waiting for that signal. And as it interacts with our target that we have brought in, BRD4, it can now transcribe through, make that RNA. And so what we've done is by using the same principles that biology uses, fuzzy logic if you will, we have 250 to 1,000 places where this molecule goes to different degrees. But if you superimpose on that the requirement that what it brings to that site will only work on a polymerase that is sitting, waiting for that signal, right? So you layer another level of specificity on there. And what you get is a single gene-specific regulator of a disease gene. And what happens in patients typically is scoliosis is the first signal that something has gone wrong. But typically people die of heart problems. You can sort of see 29 patients, majority of them have cardiomyopathies. They die of ataxias later in life. So I just had a parent in my office on Friday who told me, if you can fit my daughter's heart for another 10 years, I'll worry about the brain later. Right? So can we get to the heart? So what the student in my lab did is basically, this is what happens to the heart. So the ventricular space basically gets smaller and smaller as the muscle is trying to pump more, is building up the muscle while it's squeezing the ventricles, and very little blood goes out. And eventually this is why the patient really dies. And so these are the guys who did the work. Aska did the work where she took induced pluripotent cells. These are cells that have been taken from the patient, brought back to a stem cell-like state. This is something we can do very easily in Wisconsin because this is where some of the technology was developed. Yamanaka won the Nobel Prize for it. But for humans, this was done in Wisconsin, and we basically have access to those colleagues. And we know the signals to convert those cells into cardiomyocytes. I told you this is one of the things that we do. So she made these cardiomyocytes and then showed for certain that these are cardiomyocytes by doing PCR to make sure that you have extreme sensitivity. In fact, the cells, when they convert to cardiomyocytes, turn on the markers for cardiomyocytes, do not turn on the markers for stem cells. And the cells that we started out with, pluripotent cells, have those markers on, and not the cardiomyocyte. So this all looks good. But the cool thing here is that we treat these tissue with our molecule, and frataxin is on. And this is a tissue that's been very hard to turn this gene on using other methods that are out there. The next thing, of course, is the brain. You're losing that cerebellar region, this region that is going to give you your ability to walk, talk, and do all motor functions that you typically do. We've taken same iPSC cells, made them into neurons that represent cerebellar neurons, sensory neurons, and dorsal root ganglia. In each one of those cases, I wouldn't be telling you if the protein wasn't been made. And here, patients with different levels of repeats. This patient has very few repeats. You see that they're making, in neurons, they're making the frataxin gene to some level already. Our compound in this context is not making any more than what you would see under normal circumstances. So it does not go beyond the level that is already prescribed by the promoter. How much ever polymerase you load, that is how much is going to get to the end. You're not going to load more. So there's no danger of making too much. And this was done with Novartis, by the way. And then the other thing that we did, I was describing some of this work earlier, a year ago, to Brahmachari, who sits in IGIB. And I said, I have a colleague here who actually does ataxia screens. He's a doctor at AIMS. Patients are referred to him. He then does PCR for 20 different things and says, you have this kind of disease, this kind of ataxia. So every Friday, he collects 4 or 5 patients who have Friedreich's ataxia. And over 3 weekends, he tested 12 patients with our molecule, basically take half of it to figure out how many repeats they have. And the other half, he treated with our compound. And in each one of those cases, with the exception of this one, which was already making more frataxin than it should have been, based on the number of repeats this person has. And so what we think we've done, in a rational way, looking at the gene that we're going to target, bringing the machinery that it needs, is fix this aspect of it. What has been done in the field is this. People have tried to use histone deacetylase inhibitors broadly. That does the function to a low level, but it also has huge toxicity issues. People have tried to fix the downstream consequences. Can you use antioxidants, chelators to keep the free-floating iron, because now frataxin does not allow you to make clusters? And all of these things have not quite worked. And what we've done is we've sort of intervened at this level, targeting genomes with molecules that can target a particular sequence. And that's one path on one disease. Now, since we can make a molecule which has a DNA binding domain that you can design for whatever sequence you like, here are 40 other diseases that occur because of these repeats. We have compounds that hit every one of those repeats. And now the question is, what are you going to put on the other end to make them specific for that one disease? And we do believe that there is such a thing as precision-tailored therapeutics. I think that world is here with us today. With that, I should thank these guys who've done all the work, guys who've done all the funding of the work. But I can't resist telling you the second half of the story, how far away I am from my time. I've passed my time 5 minutes ago. Thank you. So I was hoping he would say that, so I can tell you the story properly. This was at the event that was held in Wisconsin in 2009. And it's really nice to be here. I'd like to thank the organizers. Rajendra here, Tom, of course, and Tilak Saab for repeating this. Here's Umesh talking to Dieter. Here's Gobind talking to scholars. Here are some of the scholars of the program. So these are not people who were trained by Gobind, but are Eklavya project like myself that have read Gobind's papers inspired by him. And we hope someday will become like Gobind. This is Nomura, who just iconic, and Nirenberg meeting with Gobind after many, many years. They shared the Nobel Prize for decoding the gene. This is the fellow I spent my time with who taught me to think of molecules as dumbbells. I came from a chemistry background where we looked at every atom to looking at spheres as domains. But in any case, this was the event that we held in 2009. Tom was there. And if you look at all these events, the Memorial Symposium here, the Memorial Symposium at ISC in 2011, you will find that Tom is always there, but yet he is extremely modest. He never points out that he actually is always there with Gobind. So in 2007, I went to meet Gobind, and Tom introduced me to Gobind. I had met him before, but he didn't remember me. And we talked about starting a Khorana program to honor him at Wisconsin, because most of the work that he had done at Wisconsin had really, has been the foundation for modern biotechnology, chemical biology, synthetic biology, etc. All the things that have matured into these new jazz words or buzzwords, he was already doing in the 60s. Right? So he set the stage for how you think about these problems and how you bring chemical tools and genomic tools and synthetic tools to understand biology. And so he won the Nobel Prize for this in '68, and next year will be the 50th anniversary. It's also the 10th anniversary of the Khorana program. The person who took this photograph of us is my very first undergraduate, Jay Bradner, who now runs Novartis and has developed these JQ1 molecules, a whole platform, and someday he will win the Nobel Prize, I bet. But what I'd like to tell you a little bit about, if you're not already familiar with it, that this program basically has 3 components to it. It is to provide students experience in a research lab that might transform them in a different cultural setting, to see what the horizons are and whether they can fly in that environment, what they can walk away with. There's rural development. This was brought in by other colleagues, including Gobind, and then, of course, to make it sustainable academia partnerships. The government of India joined in 2009, and if Vijay Raghavan had been here, I'd tell him how much money he gave us. But in addition to that, we had money from the University of Wisconsin to start with, and the labs that have taken on these students. This is what the students experience. They come over the summer for 10 to 12 weeks. Indian students come to labs in the US, leading labs in the US, including many of the people sitting here. And US students come back here and they go to ISC, they go to TIFR, they go to NCBS. But basically they also change their perspective on how the world works and how science is done in different environments, and the fact that creativity, resourcefulness, inventiveness exists in both environments, and how it enables different solution of different problems. Students really benefit from this immensely. But that was originally funded by the DBT, and it was limited to biology, chemistry students. So then DST stepped in and made essentially a BOSE program in 2013, so 5 years after the Khorana program. BOSE program came online, and that does the same sort of thing, but for students who are not biological. All science, math, engineering students are in that, economics as well. This is a whole cohort of students, about 100 of them, at the consulate for the orientation. And once it grew to this size, we had to start a non-profit to run it, because now what we were doing is we were engaging a lot of universities. So students choose the place where they want to go and have that summer internship. We make sure that we find the right mentors. If they have troubles with their mentors, we step in, and it becomes very difficult if it isn't within the 5, 6, 10 universities that you're familiar with. Once it goes to this scale, there isn't any major university that we haven't placed student in, and that they haven't requested students back, including Chicago. So we've done this for students. It is going really well. The nonprofit now has been able to expand the rural development component. We've worked with Vishal Bector at PAU here. We've worked with Aysar Mohali very closely. And now we're doing industrial partners like Mahindra and Mahindra to try and see how we can enable rural development at the village level. But the students, the original batch of students, now Devesh is here somewhere. Devesh, are you here? There's Devesh. He was in the very first batch, 2008. He's finished his PhD, won the best PhD thesis award. He was at Roorkee every single year. He was stood first. We're still writing a paper. He's thinking of starting an institute in Jaipur as just someone who's just finished a PhD. But his cohort and cohorts after them have graduated and now finished a postdoc, and they started this organization to try and see if they could enable people who are in the US, to come back to India and establish their careers here, or collaborate in a way that is meaningful, or develop other models where you could actually leverage the strengths of both countries and others. And Arun is here. He showed up. I just saw him. There's Arun. He's just come back as a Ramalingaswamy fellow. He was there till when? Till last year? Earlier this. So he and others were involved in building this initiative for postdoctoral fellows and senior students. And this was the first time we did it at University of Chicago in the consulate, where this team here from various universities actually came together, held this event, brought 150 people, and I think 20 or 30 speakers from India and from industry, Petroda and others. Vijay was there, etc. So this is now a recurring phenomena. We've done this again in 2017, we'll do it again in 2018. And so let me just tell you a little bit about anniversaries. So that's the report card. We've had the 50th anniversary of the Khorana Nobel Prize, well-deserved Nobel Prize, the prize that lifted the nation at a time when we didn't have this. So the impact was much greater than the award for discoveries and inventions that Gobind brought to science. We have the 10th anniversary of the program that we started in his honor to make future Khuranas, right? We have the 5th anniversary of the Bose Scholars Program for people who are like Khurana scholars but not biologically inclined. We have the 3rd anniversary of SciROI type programs, science and research opportunities in India. And what is needed is this. This man So what I'd like to propose is today we launch the project for Uttam Raj Bhandari Scholar Fellows to bring people from Nepal and the reason I'm doing it here today is because I've asked him to do this for the last 5 years and he said no. I'm trying to bring peer pressure to bear on him. So you will agree to this, Tom? Here's the team that will help you do it. You don't have to do anything, just say yes. I'll collect money from the first row. Yes?
I'll always think about what you say.
I heard yes. Thank you very much.
That was a splendid talk, absolutely splendid talk. Thank you, Asim. Yeah. So unfortunately, we do not have time because there is a poster session going on right now, and so we'll go to the poster session and there's tea, and then we will meet at 12 o'clock here instead of 11, whatever it is, 11:35. So we'll start the next session at 12 o'clock, and Matthew Sachs will chair that session. Okay, thank you.
And before going into the poster session, we have a sponsor stalls also. Please, I request to everyone to visit sponsor stalls also. Thank you.